Let me start the recording. Okay. All right. Can you guys see my screen? Okay. So hello, everyone. My name is Kim Sevilla, and welcome to my presentation. My research project looks into sperm chemoattraction and Pocilia reticulata at various stages of the female reproductive cycle. So to provide some background, sexual conflict is when the reproductive interests of males and females differ, leading to opposing reproductive strategies and goals for each sex in terms of mating. As a result, males tend to have a higher reproductive rate to maximize their fitness. Females, on the other hand, tend to be more selective and invest more in choosing mates that enable their offspring to inherit better qualities. The females can bias sexual selection pre-copulatory or before mating to select for traits such as male ornamentation or courtship. Females can also bias post-copulatory in the form of cryptic female choice, which I will discuss next. So what is cryptic female choice? Cryptic female choice refers to the process by which the female reproductive tract can influence the fertilization success of certain males or others. Pictured below is a diagram I made that illustrates how cryptic female choice works. As you can see, these three colored arrows represent sperm from three different males going to the female reproductive tract to reach the eggs. Here, cryptic female choice selectively favors the blue sperm more compared to the red and completely prevented the yellow sperm from fertilizing the egg, otherwise known as sperm dumping. These can occur through a number of mechanisms, of which the ovarian fluid presides over. So to expand more on the ovarian fluid, it is a fluid that is produced by the ovary that surrounds the egg before they are fertilized. Pictured right is a picture of me extracting ovarian fluid from a female. There's a complex mixture of proteins, enzymes, hormones, and other bioactive molecules. Its primary function is to provide a supportive environment for the maturation and development of oocytes or eggs. In addition to its normal function, ovarian fluid may also play a role in cryptic female choice by influencing sperm chemotaxis such as velocity and longevity. I'll discuss more on that later. In determining fertilization success at various stages of the female reproductive cycle. So for context on the female reproductive cycle, this is a diagram I made that represents the female reproductive cycle as having three distinct stages that is also associated with female receptivity to mating. At the immature stage, females are unreceptive until they reach sexual maturity at around 2 months. Once mature, females are receptive to and remain so until she gets pregnant and undergoes gestation. At gestation, females are typically unreceptive for about 25 days due to being pregnant. And then once postpartum is reached, the female is unreceptive for about 2 to 6 days or until she has recovered from the stress of giving birth. And then the cycle continues. Research has shown that the composition of the ovarian fluid changes with time, as noted in Gasparini et al. 2013. This may impact the movement of sperm within the reproductive tract, otherwise known as chemoattraction. Which brings me up to my next slide. Sperm chemoattraction is a type of directed movement or swimming behavior that allows sperm to navigate towards or away from chemical cues. As it was mentioned in previous slides, the ovarian fluid has tons of proteins. Of these proteins exist proteins that serve as chemical signals that direct the sperm. A majority of the sperm moves towards the egg or to sperm storages within the female. This is an important mechanism that can guide sperm for fertilization. Knowing that information and the information from the previous slides, I ask a question. How does ovarian fluid at different stages in the reproductive cycle alter sperm chemoattraction? My hypothesis is if components of the ovarian fluid changes, there will be observable changes in sperm accumulation. There is evidence for changes in ovarian fluid composition from Gasparini et al. In 2013, who found that there was a temporal decline in sperm viability when in ovarian fluid and the saline control. Evidence for the ovarian fluid affecting sperm traits that may lead to higher accumulation also comes from Gasparini in 2011. They found that sperm velocity decreases with related males, suggesting a mechanism that favors genetically unrelated males to avoid negative fitness consequences. Here is another diagram to represent my hypothesis. If sperm gets inside a receptive male, depicted here as a happy face, then there will be positive ovarian fluid facilitation, resulting in a higher sperm count in the fluid. On the other side, if sperm gets inside an unreceptive female, depicted by this face, then there will be a negative ovarian fluid facilitation resulting in lower accumulation and therefore sperm count. So our study uses males and females of the fish species Posilia reticulata. This species was chosen because it is reliable and a good study system. For methods, I created this flowchart to represent what I will be doing. First, several tanks will be set up to house and expand our population of males and females of Posilia reticulata. As babies are produced, they will be separated out and placed into baby tanks. And as they mature, they will be observed to determine their sex, and they will be removed and placed into the respective male and female specific tanks. Data collection will then begin for both sperm and ovarian fluid extraction. Fish will be placed into a knockout solution of MS-222. Also, for both sperm and ovarian fluid extraction, I'll be following the Vigili et al. 2021 and their process. Although they use zebrafish, the process is more or less the same. I will then collect sperm as well as ovarian fluid. And then to show you guys a bit of the sperm extraction process, here's a picture of a male. The structure here, as you can see, is the gonopodium, which is the male sex organ, and where I'll be collecting sperm from. For the ovarian fluid, I use a sequencing pipette to inject a saline solution to wash the ovarian fluid for collection as shown in this picture. Then a 3D printed sperm selection chamber will then be made and be used to test sperm accumulation. And here is that chamber pictured right there. And then sperm accumulation will be assessed by counting sperm using a hemocytometer. Then here's a few pictures from early testing of the hemocytometer of the sperm, and then as well as a blown up picture of the sperm. All data will then be analyzed using a two-way ANOVA using the coding language R. As of right now, we have completed the initial preparations, are able to collect sperm and ovarian fluid, and also run the chamber. But the biggest block right now has to do with counting sperm using the hemocytometer, which I'll explain in my next slide. So for my discussion, we have come across a few limitations currently and actively working on resolving these issues. We had issues with fish health regarding our species of study due to a mycobacteria infection in our populations. We have been sorting it out and have actually contained the infection to one room within our lab. We hope to rebuild our population of Posilia reticulata back up. In the meantime, we have been experimenting with other fish species within the same family and have been able to collect sperm and ovarian fluid from them. However, our process of collecting ovarian fluid is still somewhat inefficient. We are not able to recover full samples volumes consistently, but it's just one of those things that require a lot of trial and error to get the process down. But we have been able to get some full samples. And lastly, we have been having issues with microscopes within the lab. It has been contaminated from oil immersion techniques, so actually counting sperm on the hemocytometer has not been so easy and is one of our biggest issues. However, I just got word literally like yesterday of a new microscope we got. We hope to begin data collection on sperm accumulation very soon. And then I believe that continuing this research project is important because of the implications that it has on our current understanding of cryptic female choice and how it relates to sexual conflict in natural populations, as well as our understanding of the evolution of reproductive strategies more broadly. And then here are some of my citations. And then I just want to give a short acknowledgement to show my gratitude for the following groups and individuals. Special thanks to Marissa Carty, a PhD student in the Resnick Lab, and my PI, Dr. David Resnick, for their unwavering guidance and feedback throughout this project. Despite encountering unforeseen obstacles along the way, their mentorship has been extremely instrumental in enabling me to make significant progress towards my goals. I would also like to extend my thanks to Lori Graham of the Biology Machine Shop here at UCR for her help in 3D printing the sperm selection chamber. Lastly, special thanks to the undergraduate research for their financial support that allowed this project to be possible. And then thank you guys for listening, and were there any questions?
I think we'll go ahead and do the Q&A at the end.